me the opportunity to tell you about some of our more recent work. What I'll be telling you about is actually a structure function analysis of quite a peculiar ancient protein kinase containing machine that has in some way seems to affect telomere length in eukaryotes. So the work I'm going to be describing is a collaboration between my lab and this fellow Dan Durochet, a DNA damage expert in our institute. A couple of years back, Dan Durochet, actually a grad student in his lab, Mike Downey, made the discovery of an ancient protein complex that, when perturbed in yeast, gives rise to a very slow growth phenotype and gives rise to very short telomeres. So the phenotype is very similar to, or equivalent in terms of telomere shortening to a deletion of telomerase. In terms of how exactly this complex, as you'll see, is an association of 4 subunits that's very tightly connected. The mechanism by which it influences telomere length is an ongoing area of investigation in the Durochet lab, but it's still, again, very poorly characterized. And so the one thing I want you to remember in terms of biology, defects in KEOPS subunits give rise to short telomeres and slow growth. So the KEOPS complex in yeast consists minimally of 4 subunits. K1, Bud32, PCC1, and CGI121. And at the onset of our studies, our idea was to further our understanding of how this thing is working and influencing telomere length by looking at structure and the functional relationship between the subunits. So the most, I believe, central subunit in the complex is K1. It's a protein of unknown structure and function, or at least when we started. It was thought to be an enzyme, although first I should mention that this protein is conserved in all life forms, eukaryotes, archaea, and bacteria. It is thought to be an enzyme. However, there was contradictory evidence in bacteria. It was thought to be a protease, and in eukaryotes, as in yeast, it was thought to be an apurine endonuclease. And as you'll see, it's probably neither of those two. So the function of this protein was unknown. A second subunit is a member of the protein kinase superfamily. It's called Bud32. Based on sequence alignments, you can tell it's a protein kinase. It's a predictive kinase. What's interesting about this is that it's only one of 2 protein kinases that are conserved from archaea to human, across all archaea and eukaryotes. And again, the precise function of this enzyme in terms of what it phosphorylates, what it does, at the start of our study was completely unknown. The 2 other subunits are very poorly characterized open reading frames. They too are conserved in archaea and eukaryotes, but there's almost no understanding of what those proteins did when we started. And so here's our model based on knowing nothing. The fact that K1 is present in all organisms, we thought that its function, whatever it does, is going to be the key to what this overall complex is doing. And we thought that with the appearance of Bud32, PCC1, and CGI in higher organisms such as archaea and eukaryotes, we thought that these guys here are going to be doing something to help K1 carry out its function. And this model is consistent with the finding in a great many archaea species. K1 and Bud32 are fused as a single polypeptide chain. This suggested that whatever Bud32 is doing, it's going to be subservient to whatever K1 is doing. And the fact that they're all working together in an obligate manner is consistent with the phenotypes that we see. When you mutate any of the 4 proteins, you get slow growth and very, very short telomeres. The severity is such that K1, the central protein, had the most strong phenotype, and Bud32 had a less pronounced phenotype. So we wanted to probe what was going on with these proteins, first by determining the binding topology, what binds to what, then try to crystallize the subcomplexes or the whole complex if it was possible, and then hopefully we'll gain some insight into functional relationships between the proteins. So just we focus our efforts on the archaeo species because these proteins are very easy to express and crystallize, in contrast to the yeast proteins and the eukaryotic proteins, which were, for the most part, insoluble or enfolded, even when we use insect cell systems. So by fusing GST to the different subunits and doing pull-downs, we figured out the topology of the binding interaction, which is shown in cartoon form here. PCC1 binds independently to KE1, which binds independently to BUD32, which in turn binds independently to CGI121. So then we set out to crystallize the different complexes, and our first structure to be determined was this central dual enzyme core, K1 with BUD32, and that was done at 2.9 angstroms. And then we did a 3 subunit complex at 3.6, so it's somewhat lower resolution, but we're confident in the structure of CGI121 because we also solved the structure of that in isolation to very high resolution. And then we were also, our last structure, the most problematic structure due to twinning problems and whatnot, was this structure, PCC1 bound to K1, 3 angstrom resolution. We see PCC1 forms a dimer, an obligate dimer structure, and both while bound to K1 and also in isolation. So based on those 3 and a whole number of individual subunit structures, we have a model for the intact holo complex. So we were able to confirm that the binding mode that we see in the crystal environments are relevant by mutagenizing the interfaces and showing that perturbed association of the archaeal proteins in vitro, and also mutation of these interfaces in the yeast proteins caused the effect that we were expecting. Short telomeres and slow growth. So now I'm just going to talk to you about different our attempts to try and figure out functional relationships between these proteins based on the structure. And I'm going to be putting a little bit more focus on BUD32 and neglecting some of the more some of the interesting details of the other proteins. So, based on the sequence alignment alone, we had no clue what the fold class of this enzyme was going to be. When we saw the structure, we were quite surprised to see that it belonged into the ASKHA fold family of HPases. So these are bilocal structures which can support either of 2 types of functions. In some cases, they can function as conformational hydrolases, that is, these proteins can hydrolyze ATP and undergo conformational change, which affects their function. And the prototypes for this family are the HSP70 family of chaperones, that is, hydrolyzed ATP will affect the ability of these enzymes to bind and release unfolded proteins. And same with actin, hydrolyzed ATP will affect the ability of this protein to form polymers. Now, the other function that this fold supports are that allows these enzymes to function as kinases. So, for example, mainly they focused on phosphorylating metabolites, such as sugar kinase, glycerol kinase, and acetate kinase. So central to the function of both these enzymes are conserve catalytic collect at in between the 2 lobes, much like a protein kinase. This is the site of ATP binding. This is the site where all the highly conserved residues bind ATP and facilitate either hydrolysis or the phosphotransfer. So unfortunately, based on looking at a structure on its own, there's no way to tell whether our enzyme is going to be a kinase or just a hydrolase. So here's just another representation of our fold. What you can discern to some extent from the fold is one can differentiate what differentiates one protein from another in this fold class is the presence of specific inserts, and it's these inserts that give rise to the unique functional properties. So K1 has this large insert in this subdomain 2, consisting of 3 alpha units in this long linker. What's interesting about this feature is that it's conserved amongst all K1 proteins from, again, bacteria to eukaryotes. And then here's another insert that appears specific only in eukaryotes and archaea. And interesting, the elaboration of this structure here correlates with the appearance of these 3 subunits. So here's just to show you that our K1 molecule, our protein has a well-defined HPA binding site. Here are all the residues one would expect to be important for that HPase function. And when we mutate these residues, you basically phenocopy the K1 delete. This ability to hold ATP and do something with ATP is essential for this protein's function. What that, again, function is, we're not really sure. So I'll just move on to the next enzyme in the complex, and this is BUD32. We look at the structure, and in fact, it is a, it looks like a protein kinase. Yeah. So normally, a kinase has a small head and a big lower body. And what's peculiar about this guy is that the C-terminal lobe is so abbreviated, so small, that it makes it look like the head is bigger than the body. And this is somewhat unusual. So what part, the question is, well, what part is missing from the C-terminal lobe? And this is evident when this is a cross-eye stereo figure. If you superimpose BUD32 with pKa, you can see right off the bat what you're missing is the entire activation segment structure and helix alpha G, most importantly. And keep in mind, these are the structural elements that play a key role in substrate recognition. This is the part of the protein that I'm sure you're all aware that holds the phosphoacceptor site and positions it in the hydroxyl group in the exact position to accept phosphate. So that entire structure is missing. And so the first question that jumps out is, well, how does this thing recognize and phosphorylate the substrate when it doesn't have any of the infrastructure that we are all familiar with? So BUD32 is not the only one with an abbreviated C-terminal lobe. The other protein kinase in the human genome that's being structurally characterized are the rheokinases. Unfortunately, so they have the same sort of standard N-terminal lobe, except they have an additional insert on the top, kind of like a hat structure. But their C-terminal lobe is, again, very short. And unfortunately, there's no understanding of how, what precisely do the rheokinases phosphorylate, and how exactly do they phosphorylate those targets. So the question remains, how do these guys work like a protein kinase when they're missing so much of the stuff that one would expect that they need? But both of these kinases are invariant, or absolutely conserved in all archaeo and eukaryotic species. And we and us, actually Kinase and co-workers in 2007, I guess, speculate that perhaps these might be an ancestral form of the protein kinases as we know them now in human. So is BUD32 really a kinase? Maybe it's not. That's what we were thinking. Yes, it is a protein kinase. On its own, when you put it into solution, it has very low kinase activity in terms of looking at its ability to phosphorylate exogenous substrates and autophosphorylate. But when we add CGI 121, the activity boosts massively, more than 20-30-fold. In terms of does it behave like a kinase, we know that the residues in the catalytic cliff are critically important for the phosphotransfer mechanism. When we mutate those residues in vitro, we kill protein kinase activity. We're looking at autophosphorylation. So this is autophosphorylation. So when you mutate the catalytic residues, you, for the most part, lose or attenuate protein kinase activity, with the exception of the invariant subdomain 3 residue. That's the glutamic acid that laterally interacts with the invariant glycine residue in the small envelope. So this is somewhat surprising. And we know that this is the ability to transfer phosphate is physiologically relevant, because when we make the same mutations in the yeast protein and test function, we see that the phenotype parallels the loss of function that we see in vitro. And again, this turns out to be the most dispensable of the key catalytic residues. So our thinking is that CGI 121, its primary function is to bind and to activate BUD32, because the boost in activation was so robust. When you look at the structure of CGI or BUD32 with and without CGI, you don't have a lot of insight into why is it that the BUD32 in the absence of CGI is inactive. So we see some conformational changes. The beta 2, beta 3 loop undergoes like 10 angstrom shift in response to the to CGI binding. But when you look at the catalytic cleft and all the key residues in those positions, everything's sitting in pretty much the exact same position. So we're not really sure why this binding event is affecting catalytic activity. One possibility is that it's just affecting the dynamics of the n-terminal lobe structure. So we can see that BUD32 autophosphorylates. When we throw in the other subunits, such as CGI, CGI doesn't pick up phosphate. Adding PCC1, we don't see it pick up phosphate. But when we add K1, we see an interesting effect that the autophosphorylation activity gets redirected to K1, so suggesting that K1 can be a phosphate. That can be phosphorylated by BUD32. And the question is this physiologically relevant, and where on K1 are the phosphoacceptor sites? So for this, I'm just going to jump to the structure of the complex between K1 and BUD32. So based on the binding mode between these two proteins, it's not surprising that K1 can be phosphorylated. So K1 sits right on the front face of BUD32 onto a surface of BUD32 that's almost completely invariant across all BUD32 molecules. So the most conserved part of BUD32, this front face on the upper lobe and the lower lobe of the kinase, is highly conserved, and it appears the conservation correlates with composing the binding site for K1. When you look at on the other side now, K1, BUD32 clamps onto the K1 specific insert 1. So this is the surface that's highly conserved amongst all K1s, from bacteria to again to all K1 molecules. So keep in mind that this structure here predates the appearance of BUD32. So we're thinking that that's going to be critically important for whatever BUD32 is doing. We hypothesize that the binding of BUD32 to this surface might in some way perturb its ability to do whatever it's doing. So binding might in itself be exerting a regulatory effect on K1. We know again that BUD32's ability to transfer phosphate is critically important. So where might BUD32 phosphorylate K1? So recall I told you that this tail region appears to have co-evolved with the appearance of BUD32 and the other two subunits. And within this region, so this region adopts an extended conformation that winds across, spans the two lobes of K1 and approaches very closely to the catalytic cleft. So there are two highly conserved serine residues here, and we're thinking that perhaps those are the target sites. And consistent with that, if one mutates or deletes these regions in K1, you do not perturb the fold. You can still express the protein; it's still well behaved. But in vivo, the protein is completely non-functional. It almost phenocopies perfectly the K1. So we don't have direct evidence showing, using mass spectrometry or anything, that these sites are actually picking up phosphate. But that's our current working model. So I just want to touch briefly on the last subunit that I didn't mention. So PCC1, I told you in our crystal structure and in isolation we see that it forms an obligate dimer. And that just raised the question, well, why is it that PCC1 is only binding one K1 molecule in our crystal structure? And it turns out that it's just the way that we produce the proteins. But PCC1 expresses relative to K1 probably 1,000-fold more. So we think all we've done is swamped out in purifying the protein. We just did not sample the dimeric state. So with that in mind, we were able to show using analytical ultracentrifugation that if we prepare PCC1 with excess K1, that we can produce a 2-to-2 stoichiometry. And from this, we predict that this module here, the dimerization domain, and it would facilitate the dimerization of the whole KEOPS complex. So just the points that I think we're quite confident about in our studies. We know BUD32 is a kinase. We're not really, well, BUD32 is a kinase. CGI-121 is a very potent activator of BUD32 function. K1 is an ATPase of some sort. PCC1 is a dimerization module. And the way that we see these molecules come together in our crystal structures, we're absolutely confident is real. So what do we not know? So we have no idea how blood-32 phosphorylates a protein. So in the crystal structure, we don't see any phosphoacceptor sites positioned in a relative to the ATP molecule such that it can accept phosphate. So it's a complete mystery how that tail that we're seeing in K1, one could imagine that it can come loose and then go into the active site, but we have no idea how is it going to be precisely positioned to pick up phosphate. Another question, is it really a protein kinase? Because blood-32, based on its structure, is an atypical protein kinase. And when you look in the atypical protein kinase family, you see that that's where the PI3 kinases reside. So that also creates doubt. Well, perhaps blood-32, although it can phosphorylate proteins in vitro, maybe that's an artifact, and the real function is to phosphorylate something else. Another issue is blood-32 and the reals, are they an ancestral form of protein kinases? We know them now. And the other issue is, how is it that CGI 121 potentiates blood-32 catalytic function? Again, the structure of blood-32 with and without CGI 121, it's really nice when you see something in the catalytic cleft move, like helix alpha G, or sorry, helix alpha C, but we're not seeing that sort of an effect. So there's still some doubt that perhaps we don't really know how precisely CGI is affecting blood-32 function. And then probably the most important of all the issues is, what is the core function of Ka1? What is it making use of its ability to hydrolyze ATP for? And without understanding this, it's very hard to figure out the relevance of these other functions. It's kind of like a roadblock. And at least for my lab to pursue further studies. And my collaborator, the big question for him is he's trying to really, well, so what? We have all of these sort of functions, but how do these activities relate to changing the length of telomeres and affecting cell growth? Because we're not sure whether this is a direct effect or indirect. And our thing is that it might be indirect for the one reason is that these proteins, this complex is conserved in archaea, and archaea do not have telomeres. So, these are the guys in my lab that did all the structural biology and the biochemical and biophysical characterization, Dan Mao and Dante Neculi. They're both now looking for jobs. Our collaborators, this is Dan Durachet and Mike Downey. They made the discovery of the KEOPS complex. And I'm also grateful to this fellow here who helped us through FedEx crystallography to solve some of our problematic structures. So, and I want to give one little plug. So my student Tan has a poster presentation over there. Well, he'll be telling a less well-developed story, but an analogous story to what John Korean was describing for the EGF receptor. But this relates to how the RAF is activated through formation of a similar dynamic sort of structure. Okay. Does anybody have questions? John?
So, in answer to the third question up there, haven't bioinformaticists attempted to root atypical protein kinases in some kind of evolution to introduce in fact, at the San Diego Supercomputer Center, people have done this.
I think Kinase, who's here, he had a nice paper in 2007 where they were grouping all the different protein kinases, or the protein kinase-like molecules.
Why is it a question?
Because this is simply a correlation. Well, these molecules look so very different from kinases that have an activation segment, so you don't know if it's a
let me ask the question another way. Have you found intermediates? If you start from these and then fill in the gaps in each. Has that been done?
No, actually, I mean, that's one thing that we are looking at. I mean, there are obviously, intermediates possibly, but at present, there's just two distinct categories. One, or you have this alpha G helix and an activation loop that's a more elaborate component that seems to be unique to eukaryotic kinases. And then you have this more simple component like BUD32 and Rios, which don't have the G helix and activation loop. So I would it would be very interesting to actually identify a family that has, you know, maybe some components of both. So, but we haven't found that yet. So.
It's like the whole catalytic machinery is there, but it's this, I would say more regulation, this complex mechanism of regulation that's associated with the activation loop and the G loop, that whole segment is just gone. So, yeah.
The PI3 kinases have an alternate structure that allows them to hold a lipid.
Yeah.
So,
PI3 kinases are a little bit different than the atypical ones, right? I mean, they're even more.
but they still have something that gives you.
Yeah, but the choline kinase, like is in this family, that phosphorylates choline, so it's a lipid kinase. So it belongs in there. I had a question of whether, so do you have AMPP in there, or do you have any idea what kind of conformational change would take place so that hydrolysis would either mediate or facilitate binding of these other subunits?
So we were able to get structures with nucleotide, but those were even lower resolution structures. So we would just see blobs. So they were like 3.7 angstrom resolution. The way that we got our crystals to diffract to higher resolution is that we used some sort of a large cluster, heavy metal cluster complex, which was incompatible with getting nucleotide in there.
Mm-hmm.
So if we wanted to see the effect of nucleotide, we have to do it at really, really low resolution, and all you see is like a little, a blob, but you couldn't really make out precise any combinational changes.
So what happens to the association of the subunits when you have nucleotide there in solution? Do they still form a?
We see that it doesn't affect binding. But so one of the problems with studying archaeal proteins is when you study them at 37 degrees or even room temperature, those interactions might be unusually tight, so you're not seeing what's happening at the regime at which they're acting. Those experiments should be done, I think, using yeast proteins, and then seeing. The only problem is that we can't produce all 4 of the proteins from yeast. So it's hard for us to do comprehensive, detailed analysis of binding effects.
Does your kinase get inhibited by fluorosporin and analogs like that?
We didn't. So we tried making a analog-sensitive mutant of BUD32, but it doesn't work, and we hadn't tried other. We haven't profiled it against different inhibitors. We've never tested that.
So Frank, is there any condition like in the mammalian system or yeast system that K1 and the kinase dissociate?
That be pardon?
They dissociate, the complex fall apart. Is there any condition? Meaning, is there anything like phosphorylation of K1 is actually sort of leads to the dissociation? Other way to ask this question, maybe what you're seeing in your complex is a state which is, let's say, it's inactive enzyme. You know, well, there's a kinase, of course, there's a kinase, but and it's sort of, and then something happened, it ran away, and the K1 gets phosphorylated, and that dissociates. So there's a different.
Yes, we have not been able to recapitulate. We tried very hard to add things, seeing what happens after you phosphorylate things heavily. Do you dissociate things? We've never been able to see anything other than rock-type interactions. And
but that with ASKHA.
ASKHA, and then with the yeast proteins. When you run it over, when you take a yeast lysate and run it over a sizing column, you see evidence of smaller species. You see BUD32. So you see this complex travel like a 200 kilodalton mass, as if it was a big dimeric structure, and then you can see evidence of a K1 BUD32 smaller complex. We're not sure if we're just breaking it up during the lysis or whether that represents a different functional state.
I think you said you're doing this on yeast expressions.
Yes.
Can you express these in coli?
Oh, the yeast proteins in E. coli?
Yeah.
We can express the BUD32 kinase. We can't express K1, and we can't express PCC1. And our CGI 121 is precipitates. So we can produce it, and then it just doesn't last long in solution. So when you test it for function, it's hard to tell whether it's hard to make any conclusion. No, we haven't. So I was thinking that what the 2 might be for people that are interested in understanding the basics of a catalytic mechanism. But 32 might be a good model. But I would try the archaeal form of the protein. It expresses well. It's very stable.
But you're not doing
No, I'm not a Cartier NMR spectroscopist, so I was working with Julie Fournier, but her postdoc got a position at the EMBL, so we haven't done anything since our first bit of work on EPH receptors.
Frank, if you compare the archaeal and the eukaryotic BUD32s and K1s, do you see any differences, or is it exactly the same?
They're strikingly the same. So when you do projection of conservation onto the surfaces, the surface on BUD32 that binds K1 is like near invariant.
And you think K1 is a substrate of BUD32?
That's our model. That's our model. So we don't have absolute evidence. So the results are consistent with it being a substrate, but
well, if both these proteins are there across all these diverse organisms, and perhaps a function of BUD32 is solely to just phosphorylate this protein, and that's why.
Exactly. That's specialized for only. So that's why we thought that K1 would be the phosphorylation target.
Getting back to John's question, then the activation loop and the G helix presumably evolved for phosphorylating diverse substrates, and that these ancient kinases are specialized for phosphorylating specific substrates. So that's perhaps a model that we would want to consider.
Yes.
I should know what's special about archaea. Why do they express so well and crystallize so well?
I don't know. This is our first bout with Archie, and it's one of I always heard rumors of that's the system to work with. Because I guess they exist at high temperatures, so they're always stripped down, nice and tidy. You don't have big floppy loops. Thermostable.
That's true. All the archaea protein actually is smaller, is complex structure, and the room temperature at 37 degrees, they're like indestructible.
So too bad that so much of the signaling is only eukaryotic specific.
Yeah.